Hello everyone, this is S. Pavitra, a senior research fellow working with Professor Rahul Siddharthan in the Computational Biology Group at the Institute of Mathematical Sciences, Chennai, India. And the work that I'll be presenting was done in collaboration with Professor Kausal Sanyal from Molecular Biology and Genetics Unit, JNCSR, Bangalore, India. In this presentation, I'll be talking about clade identification and understanding the evolutionary trajectory of Candida auris through genome rearrangements. Candida auris is a human fungal pathogen which was first isolated from Japan in 2009 as an antifungal drug susceptible pathogen causing localized infections. It belongs to the division Ascomycota and is part of this Saccharomyces class. Candida auris is a haploid fungal species that is more closely related to the haploid and often multi-drug resistant species Candida hemilani and Candida lucernea, and it's distantly related to the diploid and clinically common fungal pathogens Candida albicans and Candida tropicalis. The difficulty in its clinical identification, multi-drug resistant properties, evolution of virulence factors, associated high mortality rates in immunocompromised patients, ability to survive high temperatures and salt concentrations, and long-term survival on surfaces in the environment make Candida auris particularly problematic in clinical settings. Within a decade, it simultaneously evolved in different parts of the world as distinct clades exhibiting resistance to antifungal drugs at varying levels. Such geographically identified clades were South Asian clade 1, East Asian clade 2, South African clade 3, and South American clade 4. And now currently a fifth clade from Iran is also identified, but it's not included in the study presented here. The mechanisms that underlie this sudden emergence, though mostly unknown, represent rapid evolution modes in a fungal pathogen. A pathogen evolves in nature to adapt to its changing environmental conditions that could help in its survival. In the absence of sexual reproduction, chromosomal rearrangements and aneuploidic changes are some of the major ways to increase the genetic diversity. In fungi, centromeres, which are the primary constructions on the metaphase chromosomes, have been observed to be the hotspots of such genome-level rearrangements. A conserved feature of centromeres is its clustering of at the nuclear periphery, thus facilitating exchange of chromosomal portions and driving the karyotype diversity and speciation. In this study, functional identification of centromeres of clade 1 was done using immunofluorescence assays for the epigenetic hallmark of centromeres, which is a histone variant called CENPA-CSC4. A closer investigation of the centromeric DNA reveals a landscape with certain common elements. This particular stretch of DNA corresponding to the centromere is devoid of any open reading frames. It does not have any functional motifs or repeats. This sequence is unique in nature in that the centromeric DNA of one chromosome doesn't align with any other centromere of the chromosomes. These centromeres have low GC content than the genome average, as shown here by the coinciding deep GC and GC3 troughs. Also, unlike albicans or Candida glabrata and more like Candida lucernea, the centromeric regions lacks pericentromeric heterochromatin. Using these features, putative centromeric locations were predicted and then experimentally tested in clade 2, clade 3, and clade 4, and also other nearby species that were used in the comparison for the study. Candida auris is a sister species to three multi-drug resistant fungal pathogens, namely Candida dubis hemiloni, Candida pseudohemiloni, Candida hemilani. These species are also closely related to another human fungal pathogen, Candida lucernea. Here the numbers 7 and 8 represent the number of chromosomes. We studied karyotype alterations in three stages. First was interclade pairwise comparison. Second was comparison with the hemilani complex, which had Candida dubis hemiloni, Candida pseudohemiloni, and Candida hemiloni. And then third was the comparison with Candida lucernea. For all these comparisons, representative images using clade 4 ISMB as the reference are shown. For the first case, genomic comparisons were performed using the publicly available chromosome-level assemblies of Candida auris to study the involvement of centromeres in clade-specific rearrangements. For the comparison between clade 4 and clade 1, no major rearrangements were observed. For clade 4, clade 3 comparison, 2 translocation events were identified. In clade 4, clade 2 comparison, 5 out of 7 chromosomes had undergone major chromosomal rearrangements, of which 3 were observed to be in the vicinity of the centromeres. Next was the comparison with the hemilani complex. Here we show the results with dubis hemilani. Similar pattern of rearrangements were observed for comparisons with clade 1, 3, and 4. But in clade 2, we observe more number of chromosomal rearrangements that are also different from the ones seen from clade 1, 3, and 4. Now, coming to the comparison with Candida lucernea, which is an 8 chromosome pathogen, the number of rearrangements is very high with all the 4 clades. But here we are also particularly interested to know about the fate of chromosome 8 and its corresponding centromere. From these comparisons, the 8th chromosome seems to have broken into 3 segments and fused with 3 different chromosomes in Candida auris. Thus, one scaffold in Candida auris would in principle be harboring 2 centromeres of lucernea, and this scaffold in Candida auris was observed to be chromosome number 4, which had Candida lucernea's centromere 2 and centromere 8. But functionally, only centromere 2 was active and centromere 8 was inactive. Though gene synteny near both the centromeres are maintained, the sequence length of centromeres, which is typically 4.8 kb in Lusitania, had come down to about 1.1 kb for the mapped centromere 8 sequence in auris. Though the same pattern of reduced length in centromere 8 of Lusitania was observed in Candida hemiloni, Candida rubus hemiloni, and Candida pseudo hemiloni, the surviving inactive centromere length showed variations. Thus, chromosomal rearrangements placed regions syntenic to Lusitania centromere 2 and Lusitania centromere 8 on the same chromosome in the hemiloni complex species, as well as in Candida auris clades 1, 3, and 4. This inactivation of centromere illustrates a clear example of evolution in progress. That is, the sequence is transitioning from a centromeric pattern to an intergenic sequence pattern. Interestingly, the case of Lusitania's 8th chromosome in clade 2 is quite different. The location of the inactive cen from Lusitania to auris serves as a useful index for representing the interclade differences. The genes in the synteny block, which were found to be on the same chromosome in the hemiloni complex and the clades 1, 3, and 4, here in case of clade 2 was found to be distributed in 2 chromosomes in clade 2, indicating that a break might have occurred within the block, followed by a downstream reciprocal translocation event. The terminal chromosomal translocation event, in which chromosome 4 and chromosome 7 of an ancestral species exchanged chromosomal lines, might have repositioned the inactive centromere, resulting in a Lusitania cen 5 and inactive centromere configuration. Now, we propose that clade 2 of Candida auris follows a unique evolutionary trajectory. Basically, Candida lusitania and other species such as Candida fructus, which is closer relative to Lusitania, might have shared an immediate common ancestor CA1 with 8 functional centromeres, 1 on each chromosome. Chromosomal rearrangements placed regions intending to Lusitania cen 2 and 8 of these 2 species on the same chromosome in the hemiloni complex species, as well as clades 1, 3, and 4 of auris, of which one was active and one was inactive. This finding indicates the existence of an immediate common ancestor, CA2. With chromosomes, chromosome number 7, with 7 chromosomes, with the configuration having 1 inactive centromere. This would have been shared by Candida auris, clades 1, by all clades of Candida auris and other species of hemiloni complex. We also hypothesized parallel evolution of the geographical clades of Candida auris at different time scales, diverging from a common ancestor CA3, which was derived from the ancestor CA2. Out of the 4 clades, clade 2 has a remarkably rearranged genome, and it consists of atypical isolates that differ from the other clades in terms of drug tolerance as well as pathogenicity. Also, the conservation of the Candida lusitania cen 8 containing the synteny block among the hemiloni complex species in all the auris clades except clade 2 further suggests that clade 2 underwent major changes in its karyotype. And these observations also prompted us to reject an equally possible alternative model, saying that clade 2 could have been an ancestral unique strain where the event leading to the chromosome number reduction could have happened. In this case, clade 2 would have then shared a higher similarity with Lusitania with respect to the synteny block harboring the inactive cen, which is not the case here. Thus, further exploration of centromere sequences and associated structural changes within a species and species complexes would yield deeper insight into the role of centromeres in generating diversity in primarily sexual fungi. Going forward, we present a rapid method for clade identification, which we call clade. Here, we want to find clade-specific genomic sequences to classify Candida auris strains into known clades. We propose the utilization of whole genome sequence data to extract clade-specific sequences for clade typing. We reasoned that some of the junctions resulting from the rearrangements might be conserved in a clade-specific manner. The PCR detection system called Clade has 5 pairs of primers. First, a universal primer pair that is designed from a DNA sequence, which would tell if the species is Candida auris or not. And second, 4 pairs of clade-specific primers designed from the DNA sequences or sequence junctions that are clade-specific. So, for the purpose of getting this set of 4 pairs of clade-specific sequences, we did pairwise whole genome alignment using NUFMER on the genome assemblies of representative strains of each of the 4 clades, and ran ShowDiff, which is also a MUMMER program, on the results. ShowDiff classifies the alignment breakpoints for the quantification of macroscopic differences between 2 genes. two genomes. Thus, we had candidate non-aligned regions for each pairwise genome alignment that could act as clade-specific sequences. We combined all these candidate regions for each clade representative genome and narrowed down the list to the ones which are commonly appearing in all the rest of the three clades, but as non-aligned regions. So, the DNA sequences from here that showed specificity in initial homology searches were chosen for designing the clade-specific primers. For example, here, one such candidate clade-specific sequence for clade 1 and the corresponding breakpoints in the rest of the clades are shown against the same position. The candidate clade-specific sequence here is a contiguous stretch of sequence only in the reference clade, that is the clade from which it has been taken, and elsewhere, that is in the rest of the clades, it doesn't show as a contiguous stretch, rather as breakpoints near an insertion or a translocation or such breakpoints. Thus, we would like to conclude that clade is fast, reproducible, and specific. We tested this method using multiple assemblies of Candida auris strains belonging to the four major clades available in the public domain, and we observed that no cross amplifications in this testing, and which suggested that this detection system is quite accurate and robust. The scope of using the primers across the strains was confirmed by testing the sequences against the publicly available genome assemblies that could include strains from all the clades using BLAST. The amplification reactions in clade described in the study are fast, specific, reliable, and reproducible. The identification strategy does not depend on DNA sequencing. The presence of the amplicon indicates the clade identity of the strain. We did not obtain any false positives confirming the accuracy of the detection system. This colony PCR-based method employed can rapidly distinguish between the four clades of Candida auris with the scope for expanding the panel by adding more primer pairs. So with this, I'd like to conclude my presentation. For references, please refer these two publications. Thank you for your attention.